So first of all, allow me to introduce myself. My name is Alex Ropelewski. I'm the Director of Biomedical Applications at the Pittsburgh Supercomputing Center. I'm involved in a number of different projects. The most relevant projects for this session is, first of all, I am the lead PI on the Brain Image Library project, which is a data archive funded through the NIH Brain Initiative. I'm also play a lead role in the HubMap consortium as well. So I would like folks to go around and introduce themselves and just mention a minute about who you are and what your institution is. Dan?
Can you hear me?
Yes, go ahead.
So my name is Dan Gabdang, and I'm leading the data ranking team at the ENCODE Data Coordination Center for the ENCODE Consortium, and I'm from Stanford.
Thank you. Joshua?
Hi, I'm Joshua Campbell. I'm from Boston University. I'm working in the HCA and the Human Tumor Atlas Network project. Within that, I'm a part of the Lung Pre-Cancer Atlas Consortium. So here we're profiling pre-malignant lesions in the lung as they progress or regress over time. So I work a lot with single-cell RNA sequencing data. I help lead the single-cell working group within that consortium.
Thank you. Anna Maria?
Yes, hi, this is Anna Maria Masci, and I'm working at Duke University. I've been involved in the first part of the LungMap project, and one of the last thing I was, I'm an immunologist, but my latest year I'm working in development ontologies and standard, and I've been involved in the last part of this LungMap project at Duke in trying to standardize the process in data coming from the RNA-seq. So, trying really to standardize all process from getting data to analysis. And that's, what I would like to try to finish and see how it could be helpful here.
Okay, thank you. Jason?
Morning, good afternoon, everybody. My name is Jason Swallow. I'm at the University of Dundee in Scotland. I run the Open Microscopy Environment Project. We build and run the image data resource, which is an image data publication system from, we're running from EMBL ABI, and we do a lot of work on image file formats and APIs.
Okay, thank you. Kylie?
Hi, everyone. My name is Kylie Degatano. I'm at the Broad Institute as a product manager, and I work with a team of computational biologists and software engineers that work on cloud scalable pipelines for the Human Cell Atlas and the Brain Initiative. And I'm here to take notes, so I'm happy to volunteer to scribe.
Great. We got our first volunteer for scribe. Thank you very much for that. Next, is it Sharmista?
Yes, hi everyone. I'm Sharmishta Ghosh-Zindigian, and you can also call me Sharmi. I am a program director at the National Cancer Institute, Division of Cancer Prevention, and I'm involved in the Human Tumor Atlas Network and the grant that Josh mentioned earlier that's focused on lung premalignancy. So I'm here to primarily listen to you guys. Thank you.
Thank you. Marianne?
Hi, I'm Marianne Martone from University of California, San Diego, and a professor in the Department of Neuroscience, and I'm part of the Brain Initiative, the Brain Consortium Data Center.
Thank you. John? John Holt, you're muted.
Oh, hi, can you hear me?
Yes.
Hi, I'm John Holt, working for Boehringer Ingelheim in Connecticut, study cancer immunology. and been doing a lot of single cell data.
Okay, thank you. And Hans. Don't see Hans anymore. Is there anybody else that I did not invite to introduce themselves who wants to introduce himself? Okay, and then I guess we've gotten all the introductions done. So first of all, we need another scribe. So can I twist somebody else's arm to be a scribe?
Yeah, sure. I'll do it.
Josh got there first.
Okay.
All right.
Joshua, you win. So
Yeah.
Kylie,
I think we could probably have more than two, so it's okay
Yes,
if you
we
want to
can.
jump in as well.
The more the merrier.
Sure.
Yeah.
Alex,
All right.
we were instructed not to have more than two scribes, right? On the intro. There were lots of directions on running these things.
Yeah, there's lots of directions, but there's the rules and then there's the practicality. So I appreciate having at least two scribes, and sometimes we miss something or somebody doesn't capture an idea exactly proper. So if somebody needs to clarify, by all means clarify. So
I'm happy to be the backup.
So in general, what we're supposed to be discussing today are some data format standards. And as we're well aware of, there's a lot of different data modalities that make these projects work. There's a lot of different ways to format and organize the data. Some methods are designed more for for the machinery that's producing the data to dump out the data quick, whereas other formats are designed to retrieve the data off of disk space, for example. And there are some community standards that exist for some different data modalities. And there are lots of areas that there are not good community standards. So let's start off by just looking at some data modalities that do not have good community standards. Does anyone want to raise an area?
I'm curious, are there any data modalities that do have good community standards so far? I don't know if I can reverse the question a little bit, but I mean, maybe I think some are definitely farther along than others, but are there any that you would consider kind of solved up to this point that we could sort of look to as like a gold standard?
Well, when we're talking about data standards, I mean, data standards is always an evolving topic. So I appreciate you're asking for some clarification here. In some ways, at least in my opinion, the sequencing world is ahead of, let's say, the imaging world in developing and adopting standards. So maybe what would be useful would be to have some input from different folks who are involved in different groups and where they see the need amongst their particular group or groups that they're working with.
Yeah, I mean, I'd be happy to start. I totally agree with you. I think the sequencing is definitely farther along than the imaging, and I'm more in the sequencing field, so I don't envy the imaging people having to think about this, because I'm sure that's more challenging. Even though the like single cell RNA-seq is farther along, I think in our consortium we're still facing some challenges And I'm curious to see if this is sort of unique to our consortium or if it's common to other consortiums as well, where our consortium has a bunch of different centers that are funded, and each center is using their own kind of platform. Some of them are, have their own preferred pre-processing tools that they use. And there was some discussion, do we try to, at the beginning, force every center to use one kind of common pipeline, even though, there's a lot of single-cell RNA-seq pipelines out there now. Do we force everybody to use one? And there was like a lot of resistance to that. So by some of the individual centers who have their own favorite way of doing things. And so, based on that pipeline, if there was one pipeline, then whatever format came out of that pipeline, you could kind of call the standard. But the different pipelines sort of produce kind of different file formats. And so instead, we worked on coming up with exactly standard formats that we can convert all the different pipelines to be sort of be concordant with each other, and then go into like our data coordinating center. So I don't know, I'm curious to see if other consortiums are similar, or if they've taken the approach of like the top-down approach of like, we're going to have this one pipeline and use this particular format for everything, no matter what you're doing.
So does anyone else want to contribute?
So for the Human Cell Atlas, we did take the approach of designing standard pipelines to process the data, and choosing standard formats. Before we started making the pipelines, we did spend a lot of time talking about which formats to use, both for accepting raw data and for producing processed data. We ended up, we've only really approached sequencing data thus far, so we ended up on FASTQs being transformed into BAMs, and said we'd consider CRAMs again in 6 months, but that was 3 years ago. So once you choose, it does kind of become the standard. And that's worked fine. I mean, obviously choosing a standard comes with its own consequences. And then one other area that I've found a bit less defined, I guess, is choosing the expression matrix format. We ended up with a ZARF formatted matrix, and it's holding also metadata and metrics just for an internal format, because we found that it supported computation more easily, but we have a service that transforms that into more user-friendly formats. So we kind of did the same, where we ended up converting.
Yeah, exactly. We just went through the process of trying to figure out that in our consortium, where it was we're trying to decide on the file format that we convert everything to. So I think we ended up on the market matrix format that 10x outputs sort of by default. It was like most of the pipelines that people were using output something like that. So that was sort of the easiest thing, except for the Optimus pipeline. We noticed that was a little bit different. So we made a conversion to convert that to the MTX format or whatever.
So does anyone else have anything to add on that front? So I do have one. Oh, go ahead.
I was just going to say, this is Jason, Alex. I mean, it's interesting listening to that. So I come at it more from the imaging side. Several of us around the table, in particular, Marianne and Alex, have had conversations. I think Marianne and I have been talking about this for at least a couple of decades, it feels like, on the imaging side. But an analogy to one of the things you just mentioned is that in the imaging world, there are some attempts to standardize, for example, just the acquisition, the metadata around the acquisition. But as soon as you move into a pipeline or a workflow, sort of one step down that path, we start running into problems immediately. And so Marianne's worked an awful lot on various ontologies that might describe the results. We've worked more, OME has worked more on the acquisition event, and, for example, defining regions of interest that would be used for profiling single cells, et cetera. But as soon as we get away from the metadata that describes a physical entity, for example, the objective lens, the microscope, it's really hard to kind of nail things down. spent some time last year or so working with Alex on a project for standardizing metadata in a specific workflow in brain imaging, and it's pretty super hard. And I think getting the workflows across all of the different steps, it seems like, is a common problem in all of these fields.
So I do have one thing to add, and you know, everybody else can feel free to also give your thoughts on this as well. You know, one of the issues we have a lot of different projects represented here, and one of the issues, if we want to, it's brought up in the plenary this morning, to exchange data. Has there been work in the consortium on defining different data states or levels for different data modalities so that it would facilitate better exchange between the different projects?
I mean, yeah, I can talk again about the single cell RNA-seq. This was actually led by some people who are part of the Cancer Genome Atlas when they were bringing up this idea of levels, and we found it really useful. So we did, I think, yeah, levels somewhat reflected kind of stage of analysis, but they also ended up just more reflecting the type of data or the data format. So like a level 0 or level 1 essentially was the metadata of the experimental protocol that was used to generate the sequencing data. And like basic information about the sequencing. Level 2 ended up being information about the BAM and like how the alignments were done, what reference was used, so on and so forth. Level 3 ends up being more of like the matrix format type stuff. So matrix of raw counts, matrix of normalized counts, yada, yada, yada. And then level 4 ends up being more like annotation type stuff, like per cell. So, you know, QC metrics or data dimensionality reduction coordinates, anything that can be fit into sort of a per cell matrix is that, you know, sort of gets put into there. So that's kind of what we decided on for our single cell sort of platform. And each one of those levels then have associated metadata with it about the algorithms that produce the matrix, and matrices, you know, you can tie different data together. This matrix produced this matrix or produced this cell annotation, and so on and so forth. Yeah, I'd be curious to see if there's a similar analogy in the imaging field or if it's just totally different.
Yeah, well, what I can mention in the imaging field, at least with the HubMap project, is that you know, the HubMap project in some cases is unique in that we have a whole lot of different data modalities but you know, just getting the consortium to the point where we can talk about data states or data levels. And I like to refer to them as data states because I know that in the genomics world, they tend to refer to things as data levels. And sometimes at data level, people think it's kind of a continuous process where you go from one level to the next level to the next level. Whereas I kind of like to use the word data states to mean this is a piece of useful information that somebody can take at that point and use it for a different type of analysis. But yeah, it is a challenge. Has anyone, you know, what other challenges might folks have with data levels or data states?
Hi, this is Sharmishta. I would let Josh add to this, but I just want to point out that in the Human Tumor Atlas Network, we are, like Josh explained, you know, we are breaking down the data into different levels, but we're doing that both for the clinical data as well as the biospecimen level data. And at some point, we have to figure out, you know, how to integrate these two. And within the Human Tumor Atlas Network, there is also, we are covering the entire cancer continuum. So there are pre-malignant lesions, there are insider lesions, and then you have the advanced cancer, and the data needs and the annotation could be very different for the different you know, areas of the cancer continuum. And for pre-cancer atlas, we know that there are still very, very large gaps in the field where we may not know exactly, how to characterize certain cell types and don't have the very specific type of nomenclature. So is there anything we can add to that, or, through this discussion, or we can consider that, in our discussion? And I can let the investigators speak to that.
Does anybody else have anything to add?
I think one thing that we run into sometimes is maybe just communicating how the data has been processed in a way that's clear enough. So for example, if we've decided that the matrices are going to have raw counts and are not going to be normalized and not batch corrected or filtered in any way, just to give all of the data, some people will come to the matrix with expectations that it will have had some of those things done to it. And it's hard to help make that clear. Oh, I think we just lost a bunch of our notes. Can someone hit Control Undo? There we go. And so I'm actually curious also to talk about, I guess maybe documentation and communication of standards, just to help people understand what's been done to data.
Yeah, this is Ana Maria. I agree with that. I think that is the thing that is really missing, and it creates confusion in this kind of very complex type of data, and where even, you can have a different approach to how to analyze data. So even if, the collection type of, what I did, I want to just explain what was the problem that we have at the first phase of the lung map where I was involved, is that we have several RNA-seq data coming from different labs, and the data were analyzed not always, using the same algorithm, not even processes in the same way. And so the idea that we were approaching was, why don't we figure out a good way to create some standard where you really annotate what you have done that could be useful to understand where are the differences, because that's, I think, is the main point. Sometimes the data are contradicting, and you figure it out that either is the processing part or is the analysis part that is not consistent between the two. So we are starting to create this, what we call an application ontology. So it means that is not, is developed to answer a specific question and problem. And in this case was about the RNA-seq, where we try to capture all the and create terms for each of the part involved, from collecting the sample to process the sample to which type of data analysis has been done. And that was, a small sample trying to figure out if something can be done at the larger level. And I think that could be done because most of these terms that I use and put together to create a more consistent representation are terms that are already present in larger ontologies. So you really do not need to create a lot of new terms, but kind of use whatever is already there, trying to put together and create this different structure that can represent the process of all the processes going on from the sample to the data, the final data.
So we have a lot of different things that we've discussed here. I'm just going to try and summarize it quickly. So there seems to be general agreement that there are no good community standards that are out there. At least nobody on this call has mentioned that we have an area where there's a good community standard. If there is, I'll give folks a moment to chime in.
I think they are community standard. For example, the ontology that I'm use, the Albo found the ontology, so mainly is the same type of format that the gene ontology use, and they are good ontology that covers the cell part, the cells, they cover the proteins, they cover the methodology for biomedical investigators, so really the techniques. And I think that that should be kind of more used. You know, there is even a good ontology for biobanking where you represent ontology of from sample, exactly how you deal with the sample coming from the process. And I think that they are not used enough, and I think they are good because there is a very large community trying to, maybe I have a bias, could be, but I think they're really good because there is a large community working on that, and they are kind of even evaluated. So it's not like that everybody goes there, put the terms, and is not control if it's good or not. So I think they are out there standard that can be used, but they are not used as much as I think I would like to see, for example.
Hi, this is Marianne. I decided to I leave my observer status and join if that's okay. I would say, and I see Amy Bernard put this into the chat window, there are some fields, certainly the initiative is really pushing standards. I think everybody knows the old joke about standards, right? That there are so many of them. And the idea that just because they're out there, that they're a standard or someone proposes them, I think there is a process that it goes through to demonstrate that it actually can serve as a community standard in this case. So I wouldn't go so far as saying that there are none out there. I think this idea that one works in a standards-based way is new to researchers pretty much, right? They pretty much do what they have at hand, what they've always done. They don't like to leave their pipelines that they've got working in their laboratories because those generally have been developed over many years. But I think at least in neuroscience, we're starting to see some standards coming in, the NWB for neurophysiology data, certainly the BID standards, the data structure for neuroimaging. Those are starting to gain traction, and you can see more and more groups are starting to use them. I am part of an organization, the INCF, where we actually developed a formal endorsement set of criteria for how, when does a standard, a proposed standard, reach the level where people can be comfortable in building infrastructure around it, using it because it's stable, it's well annotated, it's well supported by the community. And that certainly might be something that this consortium could consider doing, right? To review each of the ones that's proposed, make sure that it is usable and useful. The other thing is in formats, you want to emphasize, I think, is open formats, right? A lot of groups that we notice in other consortia, they use closed formats, they are manufacturer driven, and then you have to institute these conversion routines. And so I think having an emphasis on standards that are open standards that everybody's used is very important.
Thank you, Marianne. So just to stop for a second here, we don't have that much time left, but I want to summarize some of the ideas that folks have talked about here. So some of the areas that potentially we could work on coming up with standards in certain areas, or standardizing better so that we can facilitate the interchange of data. One was metadata. Second one was data formats, but data formats was thought to be difficult because there's a variety of different formats. Ontologies, I think that's a lot farther along, is what the discussion was, that there's some good ontologies out there that folks can use. Defining data levels for data exchange between projects is an area. Pipeline documentation for process data is also very important. Is there anything that I missed in that summary? Or anybody else want to contribute to some additional thoughts on where we need assistance?
Yeah. Can I just make one point about the ontology? I thought that was actually really, really interesting. And I can give you an example where maybe we're not as far along as we thought. At least, you know, again, I'm going back to the sequencing part of it where it should be farther along than imaging, but it's still like even what we call, even if you decide on like a standard format to put like a matrix in, there's several different versions of that matrix that you can have over time over the process of your analysis. And we don't have common names for describing those matrices. So like the original matrix that something like 10x produces is, you know, like almost 800,000 columns long, and several 20,000 rows, wide 30,000 rows, and most of those are like empty droplets that didn't have a cell in it. There's not really a common name for what that matrix is. 10x kind of calls it raw, but then other people call a raw matrix your raw counts file where the empty drops have been excluded. So some people just think of the word raw as like just a raw counts matrix. And then you can have like a filtered matrix, which maybe you exclude those empty drops, but then other people call filtered matrix when you do more filtering, like exclude on UMIs or, other things. So just even what we call different matrices at different stages of the analysis, I don't think there's, unified ontologies. At least I haven't seen them yet. Maybe they're in other consortiums too. So there could still probably be some work there as well.
Does anyone else have anything to add on that? Are there other areas? Which areas do folks think are the ones that are the farthest along, or not? Are we all in universal agreement? I'm sure there's lots of people. Everybody has their own thoughts with their own consortium. So have other people thought about data exchange? It's one of the things we didn't really talk too much about, and some of the issues in data exchange. And are there ways that standardization could help exchange of specific types of data?
I think that if you have, in order to share, you have to really know what the data are about. And we have one issue on this first part of the LangMap project where people were not using data they were on the website because we are not clear what they were about. So I think that comes along with how you kind of annotate your data, because if you don't know what they are about, it's not clear, then it's even difficult to share. And in order to share, you have to know what you are searching for too.
So can you describe a little bit more about what you mean by about what the data is about? Are you talking metadata that describes what the data is, or are you talking the way that the data was processed?
I really think both of them. So data are they have always been to describe for what they are about. So data are data, but we sometimes confuse the data and what the data are about. We most of the time talk about the same thing, but they are two different things. So the data are the number, the data, whatever we get from analysis. And then what they are about is, from where they come from, what they, how did I get the sample from what I'm talking about. So I have this RNA-seq data. That's fine, these are numbers, but how can I say what they are about? So they're about a lung, they're about a brain, or they're about a specific process, a specific pipeline. So you have to really be clear about the difference between data itself that are really, number, and what the data are about is what links the data to the reality, I would say. So in this case, it's to the biology or the process that has been done or the type of analysis that has been done.
I completely agree. Hi, this is Sharmishta. I just want to add to that I completely agree with you, Anna. And that's a full point. I mean, the source of the data and what we are comparing is very, very important. So for example, if the human cell atlas is analyzing or producing RNA-seq data from like normal lung cells, and then it kind of, you're progressing towards lung premalignancy, and you start to analyze premalignant cells, but you don't know really exactly how to annotate them or how to distinguish this transition, then we will end up comparing apples and oranges. So so we need to know exactly what we are comparing and what type of cell it's coming from, and all this information needs to be tied together. So I know that's just what I wanted to add to that I totally agree with you.
Does anybody else?
Yeah.
Go ahead.
Go ahead, Marianne.
In terms of data reuse, the word that's been used in that sense is context. We also use provenance, but it turns out, right, that the context of the data is very, very reuse it for, and that comprises everything that we just heard. And a nice term that the missing middle between the data.
Marianne, you've been breaking up.
I'm sorry.
I know that they said to keep the connection, have the video. Yeah, go off the video, and that'll give you a little bit more bandwidth.
Yeah.
So go ahead and repeat what you just said.
Can you hear me now?
Yes. Great.
Okay.
Go ahead.
A lot in various forums in which this is discussed is context, the context for the data. How is it where is it?
I'm sorry, Marianne, you're breaking up again.
It wasn't important. I'll just stop talking.
So I think what you're talking about is that the context of the data is very important, and I think that yeah, that's a good point to make.
Could I ask a question back to Anna? So you were talking about experience and exchanging data sets for the preparation of the studies that we just saw, which are really impressive. In general, so in my experience in this business, what ends up happening is someone looks at some specification, not even the standard, and says, oh, that thing doesn't represent everything I want to express, therefore I will not use it. Josh presented, an example of that where you have, a series of steps and there's just no representation whatsoever. But in the context of tissues, obviously, and anatomy, there's a reasonable amount of work that can be used. So I guess a question back to you is, how far did the consortium push to reuse the existing terminology, and where there were gaps, as there always are, I mean, that's experimental research, then say, okay, we're either going to contact whoever's maintaining this particular ontology or whatever and try to work out the concepts we want to express, or, which gets a little bit more dangerous, or actually say, okay, here's our proposal for what this thing should be, and this is where we would take it. I mean, that's always the question back, which is, to the extent to which it's possible to reuse the existing.
Yeah, no, I completely agree. And in my experience, when I started to work in the project, was not at the beginning, and so was not, I think, reused too much term from ontology that were existing, because sometimes the people do not know. That's that's the main reason when I present, I present lecture, seminar, and I talk about ontology, most of the people just know the gene ontology, for example. They do not even know about the other resources. And that is the problem. That is the problem, I think, even from the ontology part, that there is no way to, tell people that there are resources that could be useful. And so what we did, we start using what was there. And so, for example, all the cell term for the cell ontology, in addition, there's been created a new term because this was mainly on the lung developmental stage. So there were terms that were not present And usually what we do in that case is call, and I collaborated with the cell ontology a lot. You know, there is a Google Doc, you can put your term, the term request, you discuss with them, you create your definition, and you see if really is needed a new term, or if you're really, what you're looking for is something that is already there. And that's what we usually prefer, not create a term if something is there. But if you need it, you can create it. And so that was the connection, always with the protein ontology, with the cell ontology in particular, with the OBI, Ontology of Biomedical Investigator. That's where and with really Uberon, we used Uberon, and was really an interaction with them, you know, trying to reuse their term or ask for a new term. In addition, what I was trying to do, as I said, for the application ontology is something different, because that's belong to the topics that is not a domain ontology. So the term and the relation between the term I was using, so I was kind of a link, all the single part in my ontology was more specific for the RNA-seq type of data. And so I created this application ontology, but most of the term there, really a lot of the term there, I think 95% of the term there, come from the domain ontologies, just the way you link those terms that was specific for the purpose. So, and that's what really I want to do. We want to have a big ontology with, you know, domain ontology where everybody can refer to, and you can make change or ask for addition, but we would like not to have, you know, new things that everybody builds by their own, because they could be either duplication or mistake. So that was, yeah, in collaboration with the general large ontologies.
So it looks like we've got a few additional folks joining us. Maybe we'll just take a minute to go around once again and reintroduce ourselves. And so basically just state your name, the institution that you're from, and the projects that you're working with. So I'll start with myself. My name is Alex Ropelewski. I'm the Director of Biomedical Applications at the Pittsburgh Supercomputing Center. I'm the PI on the data archive funded through the BRAIN Initiative, the Brain Image Library, which is focused on microscopy data. And I'm also a key participant in the HubMap project. Joshua?
Yeah, my name is Josh Campbell. I'm from Boston University. I'm part of the Human Tumor Atlas Network project, HTAN, and I work there at the project specifically in the Lung Precancer Atlas Consortium, and I'm helping to co-lead the single cell working group there.
Matt?
Hi, can you hear me? Can you hear me?
Yeah, go ahead.
Hi,
Yes, go ahead.
My name is Matt Vichalkovsky. I'm at Washington University in St. Louis. I'm involved in the HTAN project. I'm also involved in the CPTAC3 consortium, mostly data analysis and bioinformatics.
Thank you. Shamista?
Hi, everyone. My name is Sharmishta Ghoshtandikyan. I also go by Sharmi, and I'm a program director at the National Cancer Institute and involved in a number of cancer biomarkers research program. And the one relevant to this meeting is the Human Tumor Atlas Network. Thank you.
Anna?
Yes, hi, I'm Anna Maria from Duke University. I work on the data standard and ontologies, and I've been involved in LungMap project, the phase 1, really.
Edan?
Dan Gabdank from Stanford University. I'm leading the data wrangling team at the ENCODE Data Coordination Center for the ENCODE Consortium.
Okay, thank you. Let's see, who did I miss here? Jason?
Yeah, sorry for returning late, guys. My name is Jason Swallow, the University of Dundee. I run the OME project here in Dundee. We build open source software for handling imaging data and metadata, run the the image data resource publication system running from EBI for imaging data.
Marianne?
Marianne Martone, the University of California, San Diego. I'm part of the Brain Consortium Data Center.
Michael?
Hi, I'm Mike Horwillis. I'm from the Allen Institute. Interested in the Brain Initiative single cell work, and we work on the Brain Cell Data Center with people including Marianne and Alex too. Thank you.
Thank you. Stephanie?
Hi, my name is Stephanie Morris. I'm a program director at the National Human Genome Research Institute. I'm involved in a few different programs involving technology development and also ENCODE. Thank you.
Thank you. Yunhai?
Hello, everyone. This is Yunhai Luo. I'm a data wrangler at ENCODE ETC, Stanford University, led by Tiana Pereira.
Okay, thank you. Is there anyone that I missed?
This is Tyler Best. I'm working on the HubMap program as well as ARC, which is a neurophysiology neuromodulation program.
Can people hear Tyler, or is it just me?
No, I can't.
No.
No, it's like you're talking in slow motion.
5 years of Zoom, and I've never seen that phenotype. That is crazy.
Yeah. We've seen all kinds of interesting things with Zoom this past couple of weeks, but that's the life we're living nowadays. So I believe Tyler's from NIH. He's involved with HubMap. I don't know what else he wanted to say, but there we go. All right. So I think we have another person I just joined, Tiana. Can you introduce yourself and the consortiums that you're with?
Hello. Unfortunately, my camera is not working right now, but my name is Tiana, and I'm a data wrangling intern at the UCSC Genomics Institute. So I don't have much experience at all. I was just kind of hoping I could hop on to one of your guys' sessions, maybe just see what you guys are discussing, and just take some notes for my internship.
Sounds great. Glad to have you join us. So do we still have our two note takers? Is Kylie and Joshua still on? Did we lose Kylie?
Yeah, I don't see her.
So can I have another volunteer note taker?
I can do it. This is Sharmishta. If you.
Okay, Sharmishta. Thank you. So where we left off, so what I would like everyone to do is to open up the document again that we were working on. Again, if you log on to KI Storm and you click on the data infrastructure, and then you click on 2C, that will bring up and click on the document, then it'll bring up the document that we were working on. We had a really good discussion, I think, earlier, and got sidetracked a little bit, but part of the reason why we got sidetracked is because we had such a good discussion on a lot of the different issues. And again, the purpose of this meeting is really to find out what the issues are and some issues that we could potentially collaborate on in the future going forward. So we came up with a few how we might questions at the end. And I'm going to read those right now, and we can discuss those and prioritize those in a minute here. So how might we interchange data and metadata between projects? How might we address or unify the ontologies between different consortia? How might we define data levels or states to facilitate data exchange? How might we standardize and or document the pipelines that are being used? How might we increase the findability of standards? And how might we communicate information about the standards? So with that said, are there any other questions that from our discussion earlier that we should add to this list before I open it up to the entire group for discussion? Okay, hearing none. What I would like folks to do, because there's only a limited amount of things that we can take forward in this limited amount of time, is I would like folks to, I'll call on you if you can go through and state what you think the highest priority from those how might we questions that we should discuss and address, and if there's a secondary question that you think is important, and we can make progress on it as well. And then we'll have our note takers kind of keep track of the people who had the topic as either number 1 or number 2, and that should help us determine which topics we should take forward. So again, I'm going to call on folks to state their preferences, starting with Joshua.
Sure. I'll probably, the interchange of data and metadata is probably I would say my first one. I guess the ontology one, address the unified ontology would be, I give it my second one. So maybe I'll put a 1 and a 2 next to it.
All right, thank you. Sharmista?
I think for me, it would be I agree, the interchange of data and metadata. And I would also prioritize the one that define data level states and facilitate data exchange between projects.
Okay, thank you. Anna?
Yes, so I think I agree with Joshua. Interchange data, metadata, and ontology that I think are very linked together.
Thank you. Adan? And I apologize if you hear my dogs in advance. Adan?
Yeah, I would go with the data and metadata, and the second for pipelines.
Thank you. Jason?
I'm sorry to throw a monkey wrench in here. I was thinking that for each of these, there should be a suffix or additional phrase which is in a certain domain, because I think what we're going to need are very specific domains or use cases or examples to target any of these through. But that caveat, I would agree with the interchanging metadata, and also bring up I'd add a number 2 to the findability, which is the 5th bullet point.
Okay, thank you. Marianne?
I would put my number 1 on the findability of standards if we could also add community adoption somewhere, because I think that's where we run into troubles, is finding out what is necessary to get these things adopted. So if that's kind of already included in that. And then my second one would be communicate information about data standards.
Okay, thank you. Kylie?
Hi, I'm also really interested in talking about the findability and community adoption. I'm really happy to talk about any of the other ones. And I just wanted to note that I feel like I may have gone a little off topic with mentioning the provenance of analysis and what's happening in pipelines. And there is a data QA/QC breakout tomorrow. So we could suggest that we talk about that there instead, if that's more relevant.
And then it'll just pop up there.
Okay.
So for like department, you could just, so for the department.
Matt?
You could just check the departments you want to see, but because of how the cash register journal.
Matt, are you there?
To Excel, there's extra lines that have extra stuff in it, so you just play around. I think that I would be most interested in metadata, as well as data level. It looks like I've been able to remove everything.
Okay, so if I heard that right, it was the interchange of data and metadata was your first, and define data level states as number 2?
That's correct.
Okay, thank you. Mike Holowitz?
I didn't really have, I don't have access to the document. I missed the first session, so I think I'm just going to pass on that.
Okay. Stephanie?
Hello, these all are very important. I think I would go with, as one, interchange data and metadata. Once we go through several of these, I'm going to put as 2, communicate information about data standards.
Okay. Tiana?
I also missed the first part of the session, so I don't have a preference for any of the topics.
Okay. Thank you. Tyler?
I think you commented in the chat.
Yes, he placed it in the chat.
Yeah, why don't you comment in the chat? You're still in slow motion with the audio.
Witness
please put that in the chat. The next person is Yunhai.
I think I will go interchange data metadata first, and then probably pipeline second, because I'm not sure what's going to be discussed in QA/QC. If it's go beyond QA/QC, including maybe pipeline portability, that will be also good. So I will go second for pipeline.
Okay. Myself, I would go with the interchange data and metadata as number 1, and I would go with define data states as number 2. So with that stated, there's a clear preference to begin to discuss the interchange of data and metadata. And I think that's one that we should discuss a little bit further because I think the point that that Jason brought up about, in what domain or in what, if we can narrow it down to a domain or a specific type of data or types of data, general classes of data, we may be able to make additional progress rather than just in one big group. At least that's my thought on that. What are other folks' thoughts on addressing the interchange of data and metadata?
Yeah, I want to question over about kind of what we're attempting to accomplish here over the next period of time. Is it that we're actually going to try to jump into some of these specific domains and try to like answer them directly, or is it that we're just trying to kind of come up with a process or set of recommendation that all domains should kind of go through across consortiums to answer it. So it's like if we, for each domain, we're going to recommend that, for example, there be a representative from each consortium to, get together in a working group or, something like that. And then in those, that's when like the specifics will actually be addressed.
That's a good idea. So the note takers, so I copied that bullet point down under the breakout session part 2. If the note takers go down to breakout session part 2, and let's take that note, and we'll open it up for other people to discuss things that we should be addressing in this particular area.
This is Marianne. So I also wonder, this interchange of data and metadata, is this between data repositories that serve the different projects, or are we talking about the researcher getting into the repository, researcher to researcher? Not that active in this consortium, so I don't know how this has all been set up.
I think that's an outstanding question. I think we can all answer that for ourselves. I think in general, the purpose of this series of talks is to get some discussion going between the consortium on consortium issues. So while it could be between archive and archive, ultimately it's also between researchers and researchers, so that the researchers get the data that they need to move forward. So I think both things are important, and we need to kind of consider both. One may have a higher priority than others, depending on what folks feel is the path of least resistance. So anybody else want to add to the interchange of data and metadata?
I'd just make a pitch in response to what Marianne suggested, to pick one of those and perhaps step out on a bit of a ledge here and to say, if we got exchange between a scientist or a group and a public resource sorted out, that is something that, at least there's lots of examples, they're not all perfect by any means, lots of examples, which then ends up getting adopted by a broader community. If we just do scientist to scientist, then we'll have 100 of those, like we already do. So, mind you, I'll happily plead guilty to being a little bit over optimistic on the adoption of the, if the repository comes up with something, then suddenly everybody says, oh my gosh, that's exactly.
That has been a really important, it has been an important driver, I know, at least in Brain, yeah. So I agree with you, Jason, that once you have it in the community repositories, it tends to drive the field, because you have to get your data into that standard in order to get into a repository. That leads to the development of validators and various other things then that can be used. And that's the approach that the Brain Project is taking. So I do think it's a legitimate way forward.
Yeah, I guess the challenge is that we have a bunch of different consortiums with a bunch of different repositories. So if you have multiple repositories being generated and they each choose their own thing, yeah, that's where maybe some conflicts come in. So maybe that's a goal we can try to think about how to solve here, is how we get the consortiums and the repositories talking to each other, maybe saving us a little bit of a headache downstream.
Yeah. Can you add that to the notes? I think that's a very good point. How do we get the consortiums talking to each other on data exchange?
And that's part of the community adoption too. I don't think it's just within. I think we should be talking broadly across the, whatever this consortia is, the consortia to consortia, agreements that we should be able to exchange data. And I thought the COVID talk this morning, right, showed exactly why this is important.
So I'm going to bring up another issue that we may or may not want to think about, and that is open data versus protected data for research use only.
The same thought popped into my head, and I only didn't mention it because I started to think, okay, well, if we did have another subgroup that was talking about, data access, we would then still run into the situation of the data needs to be in an interchangeable and understandable format. So either way, having a discussion on format standards seems to be productive, regardless of how soon the problem of data access is made easier, at least.
Thank you. So what are other folks' thoughts on issues with and things we need to address with the interchange of data and metadata? Kylie, did you have something to say? You're on mute.
Oh, no, I was repeating the question to myself thinking,
okay.
Sorry.
Any other thoughts on the interchange of data and metadata, things that we should or should not address?
Looks like Tyler has thrown a question into the chat. He says, how should funding agencies be involved in facilitating that interchange? Should they be incentivizing it?
That's a good point.
Marianne says.
One thing I can, hi, this is Sharmi. One thing I can say is, will it be useful to form subgroups across the consortium and, see how each consortium is doing their exchange, like knowledge and information, which is also the larger objective of this meeting, but also, talk, like find a way to continuously talk with each other and see how we can go about it. Will that, and how do we form subgroups? I mean, can we open it up to the larger consortium and see who wants to sign up and how it could be done? It's just a thought.
And I think that's a good thought too. So the question is how do we get started? How do we invite consortium to participate? And I think one of the things before we even get to this step is, we've had these talks about the different types of data the consortium are collecting with the different data modalities, but I don't think that has really been unified somewhere in let's say a page that says, or even a spreadsheet that these consortium have this type of data. I think that might help. Even if we had something like that, we might be able to facilitate some collaboration.
Yeah, that's actually a really interesting point. The first step that we did when we were working with a bunch of different centers was just to simply inventory everything that everybody was doing, what file formats they were using, and then sort of go from there. So just even getting sort of like an inventory of what different consortiums are using and what file formats and standards they're coalescing around, getting that in a central place would be a great first step.
Mm-hmm. Yeah, and I think within each sorry, go ahead, Anna Maria.
No, just saying that I agree. I think that will be a first step and will be really useful to start to see, what is out there, how the different consortia are dealing, because otherwise, it's true that you could have, I think this is the first step for communication. It would be great to have it, something like that.
Right. I was just saying that within the space of each consortium, I think we are already doing that. I know HTAN is cataloging all this information, like who has what, you know, the biospecimens, the technology that we're using. And it's just finding a way to communicate across consortia and make that available to each other.
I think that's an excellent, very concrete thing that could be proposed moving forward. And I think going down to the level of the file formats and other things, you know, the standards used, there should be, what you're telling us already, some work that's been going on within a consortium, and now we can do it cross consortium just by.
Right. It
Yeah.
should be straightforward, exactly, because we've already each done this internally with our own working group. So it's just a matter of a mechanism to share that with others and getting some key people from each consortium involved to just sit down and kind of look at it and discuss it.
Yeah, and keep it up to date and so on and so forth.
Exactly.
One other thing that has been quite a topic of discussion in HTAN, at least at this point, is coming up with data models that are used in order to store this data. So I think in addition to looking at the data formats and the metadata, I think that trying to understand better the data models that are being used across the different consortia might be useful. Having compatible data models is very important to be able to share them easily across the different systems.
Can you describe a little bit more what you mean by data model?
Well, a data model really is just the collection of metadata that you're holding on to. There's different sort of standards or different sort of approaches that we looked at when we were thinking through the data models that will be used for HTAN, for instance. As a specific example, the GDC, the Genome Data Commons, has a particular data model, and we're investigating whether that's a data model that can be applicable to something like HTAN. I know that there are, I know that we're developing other, for HTAN, we're developing other data models as well. And, you know, things like, for instance, are you keeping track of where in the body the tissue is obtained? And, you know, whether two pieces of tissue might be adjacent to one another. If one data model includes that, in other words, if that metadata is captured in one system and it's not in the other, it's important to know that because these might be real barriers to being able to interchange data easily.
So do you think that potentially the group could come up with best practices for either collecting data or for analyzing data, or is that a bit out of scope for what we're talking about?
Well, I think as a first step, maybe just having an understanding of the sort of metadata that's being tracked for specific types of data across a different consortium might be very useful. I think analysis is another another important consideration. That's something that I think a lot about myself, but I think that just collecting, having an understanding of how people categorize and the dictionaries people are using across the different consortia might be quite useful.
Yeah, that could lead to, you know, I see what you're saying. To facilitate data exchange, you need to, sometimes you need to do this translation and understanding, yeah.
That is sometimes useful even inside a consortium when you have different data type inside the same consortium. Like, you know, if you have RNA-seq data, you have imaging data, you have flow data, you have microscopy data, or all this. So I think that's, even inside the consortium, is going to be a problem. At least we have that kind of problem.
Right. There's a lot of energy being spent right now inside the HTAN consortium in order to develop these. We have a process, and it's the iterative process really, that basically we crowdsource the sort of information that people are capturing right now and that they think is important to capture. There's some sort of process by which people have a conversation about this that's then frozen for one version. what's anticipated in the future is that this is going to be an iterative process, and we might have version 2 and version 3 of metadata. And so we might actually ask the different centers, we might go back to them and say, hey, the data that you uploaded, back in December, can you please provide additional information with that as those metadata standards get evolve. So just based on that experience, how it is an iterative process, it's quite important, and it's difficult to impose either from the top down or from the bottom up exclusively. Given that even within the consortium, it's a important but relatively difficult thing to do, at least having an understanding of what the other consortiums are doing, if they have kind of converged upon a set of metadata standards. I think it would be good just to catalog that.
Okay. Does anyone else have things to add on the interchange of data and metadata? Some of the things that we should address or consider?
I'm not a oh, sorry.
Sorry, Kylie. Just quickly, just to respond to the last 2 points. Obviously, it makes great sense across these consortia, HTAN, HubMap, or whatever, to catalog that constellation, shall we say, of different formats. Does anything happen after that? I mean, at least in the world that I know, in imaging, we're dealing with a pretty wide variety of different formats. Can you use that to narrow, in practice, narrow choices or narrow the diversity? Or
I can speak in one consortium I'm involved in. It's not part of this consortium, it's part of another consortium. But we, the first thing we did, because we were getting such diverse and heterogeneous data in, is we automated and said, what are all the file extensions that are used? And there's 140 different ones. But you also saw that they broke down into categories, and based on that, we said, okay, these are the ones we're going to accept, right? Because you saw ones that we did a little analysis, which ones are in wide use, which ones are open. We had a set of criteria. And so we're going to start enforcing and imposing those now. So I do think I think it was an eye opener for the consortium, just how bad the data were, right? And how many proprietary formats, custom formats, people were using. So I do think if there's an action that can be done with this, and I don't, the governance of how this is going to work, I think has to be discussed, but I at least found that that was the first step towards getting some traction rather than uniformly just declaring. We said, these are the most common used, these ones will be good enough because there's a lot of converters. So yes, you're allowed to use this particular format.
Great.
So I think,
Go ahead,
I just wanted to put down one point here that I think you raised that we haven't discussed this far, and we can continue your thoughts here. But you did mention governance, and I think that's also very important. If we're going to go down this path, how do we foresee coming to agreement on different issues that we need to come to agreement on? But go ahead.
Yes, this is Sharmi. I just wanted to add to what Jason already, I think, alluded to, which is I think maybe we are talking about data harmonization across the consortia, if and when possible, because our areas of interest are also very different, right? Some are working on normal cells, and then some are working on tumor cells. So, and within tumor, we have like different types of cells that we are working on. So are we talking about building common data elements across consortia, at least to have the capacity to have a common data ecosystem, which I think is the larger objective of this meeting, and be able to do some cross-consortium analysis, or is that too ambitious or difficult at this point?
I think that's a very good question. I think it's useful to answer, do we really want to be able to share data across the different consortia? And then what the scope of that sharing is. Maybe thinking through some use cases or examples where that might be useful. But I think if we do decide that we do want to have the ability, in principle at least, to share data across the consortia, then I think we have to make sure that we speak the same language, and that, you know, if I ask for something, at a minimum, that I know what I'm looking for and what I can expect to obtain.
Yeah, and I can speak at least from the hub map perspective. Is what we're collecting through hub map is healthy tissue, and you can define healthy, you know, a bunch of different ways because nothing's really 100% healthy. So with hub map, anyhow, we're not getting the diseased tissue, but you know, the ability to compare healthy tissue with diseased tissue, I think is very powerful. So I think some consortium will have more of those issues than others.
I wanted to ask, does anyone know if any group in the GA4GH is thinking about things like this?
Yes. Almost every consortium is thinking about this. And I was kind of chuckling to myself because use cases and catalogs, we may as well just start with those because that's what everybody converges to. But all big consortiums or small consortiums are trying to grapple with these issues. And the question is, do you solve it within a consortium? This is a little more ambitious because it's saying we don't even have to solve it just within, but also across. But I think, I bet you that as we were saying in the chat sidebar that almost every group is coming to terms with this, right, in some way or another, right? What are the common things across? How do these things get governed? How do decisions get made? So
right, I was kind of hoping
a lot of good material though that can be used. Yeah.
Yeah, and I was hoping that maybe since the GA4GH is more aimed on standard setting, that they could be leveraged or worked with, or a group could be joined to kind of help facilitate.
I agree. They've done a lot of work, and in at least a recent talk I heard from them, once you get to the fact that you can exchange data, then you get into the QC and the validation as to whether data exchange across consortia can actually be compared. So it starts to lead to you know, additional levels of interoperability.
So one of the points that Tyler had mentioned is how should funding agencies be facilitating data interchange and I just wondered if anyone had any thoughts on that. I mean, there's a lot of different models that are used. You know, should things be more driven by the agencies? Should there be, you know, should it be grassroots where we just come together and then hunt for a way to make things happen? I mean, obviously with a lot of these things, you know, money, we all need money to be able to devote resources and take resources away from our individual projects to solve the larger project. So I just wondered what folks' thoughts are and what might be the better way to handle that.
I think this is Sharmi. Since you know, I'm from NIH, I can just see that. I think for the investigators, maybe it will be useful to first identify areas that are important, you know, cross consortia, and that we can work on together. And one is the coordination part of it, and then the collaboration part of it. And of course, there are, you know, financial resources that are involved if something tangible develops. But I think it will be useful for us to first know that this will be useful, you know, something that we can, it's doable, you know, across the consortium. And what are the areas, like we're talking about, you know, to build a larger data ecosystem, what are the areas we need to focus on, and how can we communicate with each other, you know, what would that entail? And then we can go from there once we have that information filtered, you know, through the discussions at this meeting. That's just my thought.
Does anyone have additional thoughts?
Sorry to be provocative again. Can you define useful? And I mean that and I mean this in a kind of serious way, because I mean, obviously there's been a lot of discussions about standards, and Marianne mentioned, consortium looks around and says, oh my gosh, we've got 140 different file formats. That's a pretty difficult place to start. So,
Jason, sorry, I missed the first part of your question. Sorry,
no problem.
The audio connects.
So it's probably my fault. I just asked, can you define useful? What would it mean to be useful?
Oh, okay. I mean, what I meant was, like we are talking about, the different areas that HubMap has. They're doing, studying, single cell analysis across disease types, or human cell atlas, and then human tumor atlas is doing across different types of tumors. Do we see common ground across these different consortium? Is it like, for example, is it technology oriented? Or should we be focusing on something that, single cell or single nucleus RNA sequencing that this different consortia they're doing? Maybe the larger objective is different, but there are common grounds that tie us together. And can we filter out those information, and then we can form subgroups that we can work on? That was kind of what I was trying to say. Does that make sense?
Yeah, that makes sense. And I think it's even linked to what we said before about how each consortium is dealing with their own data and metadata. The thing that Joshua proposed before, to know exactly how they're dealing. So if we can start from that and know what each consortium is going to do and which are the common thing that could lead to what you're saying, that could be, I think, one point, because it's true, it needs to be coming from the user and then go, it should be a collaboration with the agency too, because unless you have to find the problem from the user point of view, where the people are dealing with the data and using the data and say, we need to, when I look to this consortium, I found something and different in this other one. So here are my problems. But then it needs to be kind of pushed out and enforced by the agency, because then otherwise it's not going to, to go anywhere.
Right.
Yeah, just to follow up on that, I'm wondering, I'm scribing here, and I want to just think about what to write down underneath the recommendations, what we want to do, and just try to be specific, as possible related to that point. So, I mean, we've had discussions about trying to get people together across consortiums to, exactly, inventory and start addressing these points. Is that, I mean, do we want to make that as like a specific recommendation, that there be some sort of committee or working group, cross-consortium working group formed, I guess maybe for each independent data modality?
Yeah, I think the first step would be really to do that working group, but I'm not sure doing it by data modality would make sense. I mean, I think the first priority seems to be to get the inventory of what people have, and I'm just not sure that, we would want to do that by data modality, but I don't think we would necessarily need data modality experts from each consortium and have a lot of people, working on this inventory gathering at the same time. Does that make sense?
Yeah.
So I think at some level, it's really identifying the folks at the different consortium who know their data and can provide input into this, whether we call it a spreadsheet or a document that we're developing through this process. Does anyone else have thoughts?
Just wondering for the topic of funding agency driven use case, I'm wondering, is the funding agency for the consortium or also include funding agency to a data producer?
I don't know.
I mean, what I'm thinking is like, I feel like there's a difference here based on my limited experience, honestly. I mean, is that we are ENCODE consortium, we have our own funding source, NIH-HCA, and they want us to have standards. They want to everything in a good shape, formalize it. But after all, we are a data coordinating center. We're not producing data. It's really the lab who's doing this hard work, wet lab experiments produce those data. And then they potentially have projects funded by other funding agency. And one problem I personally saw is that when they generate the data, they want to publish, and they deposit data to either us or to some other repository. And what they will find out is that, okay, the funding agency doesn't care too much about that, and I would just deposit to I probably shouldn't name anything, but just deposit to a repository that doesn't require too much metadata to collect. I mean, that's easy, it's simple, and I'm not going to deposit to, say, for ENCODE, which collects a bunch of metadata, take my students so many times to, formalize it and then upload them. So it's really, okay, we have data model here, and we have a validation here, data in good shape. But if the data producer have no, I don't know what's the correct word, no strict guideline or anything, encourage them to submit to a repository and collect metadata, they may just go, okay, there's a repository out there, just accept files, and then that's it. I would just put my FASTQ file onto it, and then done. I have publication. And so that's, I feel like potentially that's what I saw the difference between the funding agency for data producer versus funding agency for consortium, because funding agency for consortium, they know the thing here, they care about data model, data standard, metadata, interchangeability, all this FAIR principle. But for wet lab, they're focusing on the science and everything, which they did definitely the right job, but they have the data, right? So if they don't submit metadata to us, we don't know what to do, I guess.
So a clear policy statement from the funding agencies would be helpful in that regard?
I guess I'm trying to go somewhere along that line to say is that, we're a data center, we cannot push, okay, upstream production lab, please collect this metadata. We didn't fund them, right? So we're not their funding source, so yeah.
I think one thing I can say is normally what we do is we develop data use agreements with, if we are sharing data across consortia and people review that, they comment on that, they agree to that, and they kind of like they're bound by that data sharing, agreement document that they have signed off on. But, I'm sure there will be a lot of discussion and bumps along the way, because at what level do we want to share? How do we do that? And, some people may not agree to that. Would it be sharing of raw data files? Would it be, curated data? And all these discussions, it gets into, complex areas, and then we have to sort all those out if we are doing that. That's, I don't know if someone else wants to add to this.
Hi, this is Stephanie. I think I would probably say that what you're talking about, not you, Sharmi, and I'm sorry, I didn't catch the last person's name that's involved in ENCODE, seems very consortium specific, what's being deposited with the coordinating center versus what other support the data producers have and their requirements for those other types of data. And I would probably say that it's not, each institute has things that are specific to the individual consortia, but it wouldn't just be NHGRI. This would be more along the lines of probably NIH thinking of how they can support some of these efforts that are really in response to the needs of the community.
Yeah, and I think that is true because if the people that are producing data are not incentivized to give all the information, they will never spend time in doing it. But on the other side, I think that sometimes the consortium, the data repository needs to have implement a lot, which is the uploading of data kind of way, because sometimes it's too complex or is not exhaustive from the side of the researcher that is uploading the data. So I think that is even another point that maybe the consortium should be thinking when and should be getting funding to kind of facilitate it and the uploading of data at the same time get as much as information about the data, and if it's possible, an automatic way to annotate the data that are coming in.
Okay, this has been a great discussion. We have about 20 minutes left. I think what we should do is, if we go to the document, the blue section that's starting to be filled out, I think this is the appropriate time for us to review this. And I think, Joshua, you've been taking a lot of notes here. So if you want to take the lead on what you've entered here and go over it and what the action items would be, and we
sure.
can.
Yeah, I think.
What else we need for this document?
Yeah, sure. So I think Kylie also took a ton of notes here. The blue box, I guess the beginning of the blue box underneath recommendations is just some additional sort of notes, but I tried more towards the end, I tried to make this concrete in the, I guess, action items, or maybe a sort of suggested procedure that we should go through. So maybe if everybody could focus on that, and we could just clean that up so that way we can present that as something moving forward. So the first action item was each consortium should identify individuals or a group of people to perform an inventory of data formats across all modalities. And this should be sort of deposited in some sort of central location, even just a spreadsheet or something. Second action item would then be to form a cross-consortium working group, likely with the individuals from point one, who sort of get together and examine this central inventory, and we can determine sort of how similar or different the data standards are across each consortium. And maybe, I guess that's probably where the real work would take place, deciding on, are there some formats for each modality that we want to sort of officially report support. That's it. Let's say that they can get to some sort of consensus, then we might want to make recommendations to the funding agencies, who then they can officially sort of support and push for these formats across new and more importantly, existing studies, and more importantly, new studies. So that way everything going forward can then already be, sort of adhere to these standards. And then maybe, I only listed data formats and sort of in the first 3 points. So you could either repeat the process for like the metadata and data models, or you could maybe do it alongside of the data formats. I don't know if people have specific preferences there on recommendations, but that's what I have right now. But yeah, please edit it, or curious to get people have other thoughts.
Are we saying though that so the form of cross-consortium working group will be the sort of group that does this work, right?
Yeah, ideally.
So I'm wondering if that should be number one, because that would be the vehicle, and then each consortium should do this and put it in a central location determined by the group, right?
Yeah, 1 and 2 are kind of related, I guess. So you can put them all underneath one if you wanted to. Yeah, because the individuals doing the inventory would likely be the individuals in the cross consortium working group. So
I just switched.
That's fine. Okay, got it. Will do.
It sounds like a lot of folks from the data coordinating centers probably will play an important role in this, right?
Yeah.
Everything filters through them.
That's a good job on those notes of an action item. Joshua.
Yeah, sure. Oh, good.
Josh, reading numbers 1 and 2 on the action items, which are nice summaries, it kind of seems like, and maybe this is already done, but it kind of seems like there's another kind of 2A or something, which is to publish the existing inventories and the overlaps between the different consortia, just to make it very, very clear, so that effectively everyone in the consortia can see where the opportunities are for those connections or overlaps. Is that already done?
Yeah, I think that's a good point to add, that we can add like what's going on right now, where somebody's adding the sub points. I think that's a good way to handle that. I think we should also address how we get people, I mean, we have this initial group here, I'm assuming, with people who've added their names and the consortium that they can assist with at the bottom. The other real question is that how do we get other consortium involved in this process? It's great to start out small and then grow.
Mm-hmm.
Do we have enough key consortium folks on this?
Yeah, I mean, it's a good point. We sort of need at least one person from each consortium. So at some level, I don't know if this means going to the program officers or, the people in charge and like saying, here, find that person, or, so maybe there should be like some sort of top-down assignment, and then they can start working on it.
Yeah.
Should we put that down under additional expertise that we need?
Mm-hmm.
And say something along the lines of, I don't know, we need a person who's familiar with the data on each major consortium to be represented.
At least somebody who would be funded to go and try to ferret that information out and be in contact, because it's often a lot of work across these consortiums to figure this out.
Is it fair to say that every consortium has a data coordinating center? Is that a standard thing?
No. Well,
well, that's the term that tends to be used with folks who are doing a lot of the genomics projects. With projects that aren't genomics related, they have a similar function, but you tend not to hear the word DCC.
The BRAIN initiative has a coordinating center for the brain cell census consortium, the BICCN, but as far as I know, it doesn't have it for the rest, which is distributed across multiple agencies and involves a lot more of NIH than just the BICCN. So that would be a real investigation to try to pull that information out. NIH has some materials on it, but again, you'd have to go into NSF, you'd have to go into a lot of places.
Yeah, I mean, the other issue too with BRAIN is that BRAIN's organized a little bit differently, in the sense that there's data archives that are focused on particular data modalities.
Mm-hmm.
So, with some of the data modalities, you'd be coordinating with one archive, and with others, you'd be coordinating with a different archive. So there's also these organizational issues that we would need to look at.
But I think for BRAIN, we would just focus on the consortium part of it, because this is a consortia effort. So I think we would just do the BICCN. Because that has a governance structure and something that can speak on behalf of the consortium.